So, very good morning all. Today we are going to deal with the chapter respiration in plants. Okay. As yesterday we have discussed the photosynthesis in higher plants in connection with nutrition in plants of 10th. Today we are going to deal the continuation of that is respiration in plants. From this, in every year NEET, definitely from this chapter you will get definitely 2 questions for sure, only from this chapter of plant physiology. Fine ma. So, first thing I would like to tell you that yesterday you have clearly observed how the primary product of photosynthesis is formed. What is the primary product of photosynthesis beta? Glucose. Glucose is formed where? Inside? Inside? Chloroplast, right? Stroma of the chloroplast. What are these? Membranous sacs. This is stalk of sac like structures. What is it? Grana. And this is fluid filled space. Inside a stroma what is formed? Glucose. Glucose, to store, it gets stored in the form of starch. Okay? It gets stored in the form of, but it has to be transported though. It will be transported in the form of sucrose. Clear beta? Okay? Glucose which is formed at the end of photosynthesis, if it has to be stored, it will be storing in the form of starch. If it has to be transported, it will be transported in the form of sucrose. Okay? So, now the food material is prepared. Okay? Some material is being stored, some is being transported. Why the food is prepared actually? To get energy. Okay? Why do we take food? To get energy. What is respiration in detail? Can anyone tell me? What is respiration? And what is breathing? What is respiration and what is breathing? Is there any difference between that? Sit straight, be attentive, listen. Breathing is nothing but exchange of gases, right? What is breathing? Exchange of gases. What is respiration? Oxidation of food material inside the cell is called as respiration. What can be the oxidation of macromolecules? The macromolecules can be anything. Okay? The macromolecules can be anything, be it carbohydrates, proteins. Tell me some more macromolecules or food materials that we take. Proteins, fats, yeah, fibers, everything. Okay. So, what is respiration? Oxidation of macromolecules. Fine. And all these are called as what? Respiratory substrates. What are these called as? Respiratory substrates. Fine. And what is cellular respiration in detail? Okay. What is cellular respiration? Oxidation of macromolecules. Okay, oxidation of macromolecules inside a cell and release of energy in the form of ATP inside the cell is called as cellular respiration. What is cellular respiration beta? Oxidation of macromolecules or oxidation of respiratory substrates inside a cell and release of ATP or the energy inside the cell is called as cellular respiration. Okay? And ATP is called as what? Cellular energy. ATP is called as what ma? Cellular energy. So, are you clear with these points that glucose which is prepared in the chloroplast is stored in the form of? It is transported in the form of sucrose. And what is respiration? Oxidation of macromolecules. And those macromolecules we are calling it as respiratory substrates. What is breathing? Exchange of gases. Clear with these terms beta? Cellular respiration means oxidation inside the cell and release of energy inside the cell. What is cellular energy? ATP is the cellular energy. Clear with this, right? Now, do plants breathe is the question from this diagram. Do plants breathe? Do plants breathe? Okay, this is the question. Okay, how do plants breathe, or do plants breathe? Answer me. Yes or no? Yes. How? Yeah, exchange happens. How? What are the respiratory organs? Are there any respiratory organs in the plant? No, right? They have the organelles like, stomata in the leaf, lenticels in stem. Okay, stomata on the leaf and lenticels on stem. They will mediate gaseous exchange. Gaseous exchange. Okay, stomata on leaf surface and lenticels over the stem, they will mediate what? Gaseous exchange. And not like animals, plants will have great respiratory needs, because all the cells of the plant organs will take care of their own respiratory needs, and there is no gaseous exchange demand by all the cells. Fine. And just now we have discussed about transportation of glucose in the form of sucrose. Of course, for example, in a plant, over the leaf glucose is formed, but energy is required for root cell also, energy is required for shoot cell also. Okay. So, the transport takes place in the form of what? Sucrose. Now, we will see how the respiration takes place in plants first, and then we will see how anaerobic respiration also takes place. How oxidation of macromolecules occurs in different cells, we will look into. Okay. So, now if you have to see, respiration occurs in two ways. One, in the presence of oxygen, and one in the absence of oxygen. Okay. In the presence of oxygen, it occurs in the higher plants and animals. Plants and animals. So, what happens in both the aerobic respiration here, which is aerobic, and in the anaerobic respiration, one process is common beta. What is that? In both aerobic and anaerobic respiration, one process is common, that is glycolysis. Okay. Glycolysis is common both for aerobic and anaerobic respiration. So, what is glycolysis? Glycolysis is nothing but, glyco means what, Nana? Sugar or glucose you can consider. Lysis means what? Breakdown. Okay. Partial or incomplete oxidation of glucose is called as what? Glycolysis. It is also called as EMP pathway, which is named after the scientists Emden, Meyerhoff and Parnas. Okay. It is also called as what? EMP pathway. Fine. And this is incomplete or partial oxidation of glucose. For example, if you have to consider this as a cell, okay, and this is the cytoplasm and this is the mitochondria. Okay. Glycolysis happens inside the cytoplasm. Fine. Sucrose has reached, for example, let us suppose from leaf cell, sucrose has reached to a root cell. Okay. Once after reaching the cell, sucrose first breaks to, okay, glucose and fructose by the enzyme invertase. By what enzyme, ma? Invertase. Sucrose will break to what? Glucose and fructose by the enzyme invertase. Now, glucose will start the process of what? Glycolysis. Okay. At the end of glycolysis, one molecule of 6 carbon glucose will break to 2 molecules of 3 carbon pyruvic acid. Okay, 2 molecules of pyruvic acid. This pyruvic acid, if it is conducting aerobic respiration, this directly lands to the mitochondrial matrix. Where does pyruvic acid go? Directly to mitochondrial matrix. In mitochondrial matrix, the very first step after glycolysis is oxidative decarboxylation. Okay. Second is TCA cycle or Krebs cycle. Fine. These two occurs where? Inside the mitochondrial matrix. And last step, that is electron transport system or oxidative phosphorylation, happens in the inner mitochondrial membrane. Okay. So, here this is clear to you know beta. In aerobic respiration and anaerobic respiration, what pathway is common? Glycolysis is common. Glycolysis is also called as EMP pathway. This is an incomplete or partial oxidation of glucose, wherein sucrose after entering the cell, by what enzyme it is breaking down? Invertase, it breaks down to glucose and fructose, where glucose will be converted to 2 molecules of 3 carbon pyruvic acid. Pyruvic acid will directly enter into mitochondrial matrix. So, the first step is glycolysis, second in the aerobic respiration will be oxidative decarboxylation. Remember the steps ma, oxidative decarboxylation. And third is, after oxidative decarboxylation, what is the third step? Third step, it is TCA cycle, okay, or Krebs cycle. What is that? TCA cycle or Krebs cycle. Fourth, last and final step, what is that? Electron transport system, which is activated in inner mitochondrial membrane. Both these, oxidative decarboxylation or link reaction also we will call it as, these two happens to be where ma? In mitochondrial matrix. So, are you clear with the 4 steps of aerobic respiration? The very first step is what, Nana? Glycolysis. Second is oxidative decarboxylation. Third is TCA cycle. Both of them happens to be in mitochondrial matrix. And last step is electron transport system, that happens in inner mitochondrial matrix. Clear with the aerobic respiration steps, right? No doubts anywhere, right? Now, anaerobic respiration, if you have to see, in anaerobic respiration ma, everything happens to be in cytoplasm only, because this happens mostly in the prokaryotes and no membrane bound organelles will be there, right. So, everything happens to be in where? Cytoplasm itself. And in anaerobic respiration, after glycolysis, based on the cell location and type of fermentation, we have 2 types. One is lactic acid fermentation and second is alcoholic fermentation. Okay. First is what ma? Lactic acid fermentation. Second type is what? Alcoholic fermentation. So, today in this class we are going to deal in detail about one process, that is glycolysis. Common process both for aerobic and anaerobic respiration, and we are going to learn in detail both lactic acid fermentation and alcoholic fermentation. Clear ma? Don't be drowsy early in the morning. Focus. Now, so the very first process, what is that? Glycolysis. I told you one thing, right? Glycolysis is also called as, very good Aisha, it is EMP pathway. Here in glycolysis, glucose is completely oxidized or partially oxidized. Partially oxidized or incompletely oxidized we will call it as, okay. Partially, or remember these terms, more important, partially or incompletely oxidized. Okay. Glucose is how many step process? Glycolysis is how many step process? 10 step process. Okay. Glycolysis is a 10 step process. 10 steps of biochemical reactions happen, over which the first 5 steps are energy utilizing phase, okay, energy utilizing phase, and the rest 5 steps are payoff phase, or you can also call first 5 steps as energy investing phase, next 5 steps are what? Payoff phase. over that the very first step is what ma? Glucose to glucose 6 phosphate, where it is utilizing. Yesterday I have told you know how you have to understand by looking the reactions. Here ATP is utilized because it is over here to the reaction. One ATP molecule is utilized from conversion of glucose to glucose 6 phosphate. This first reaction is catalyzed by what enzyme? Hexokinase. Okay. Next, glucose 6 phosphate to fructose 6 phosphate. Okay. Glucose 6 phosphate to fructose 6 phosphate. This is by what enzyme? Phosphoglucoisomerase. What is the enzyme ma? Phosphoglucoisomerase. Third step, which is the rate limiting step of the reaction. Okay. Third step is what? Rate limiting step. From fructose 6 phosphate to fructose 1,6 bisphosphate by the enzyme phosphofructokinase. Okay. Again here one more ATP is utilized. Clear? Fourth step is fructose 1,6 bisphosphate to, it splits to glyceraldehyde 3 phosphate and dihydroxyacetone phosphate, which is DHAP. Okay. This is mediated by the enzyme aldolase. Okay. And here you will get 3 carbon and 3 carbon, till here it is 6 carbon, right. So, here they can be isomerized. From here the rest 5 steps can be continued with glyceraldehyde 3 phosphate. Okay. Rest 5 steps are continued with what ma? Glyceraldehyde 3 phosphate. Once these 5 sets are done, then DHAP will be isomerized to G3P and the next it will be continued. So, from here we have to calculate for 1 glucose, not for 1 G3P, okay, because the first step started is from 1 glucose, understood. So, here the enzyme which is isomerized is isomerase. And glyceraldehyde 3 phosphate to 1,3 bisphosphoglyceric acid. This is by the enzyme glyceraldehyde 3 phosphate dehydrogenase. Fine. And here the energy released is 2 NADH. How much ma? 2 NADH, because we are calculating it for 1 glucose. Next, 1,3 bisphosphoglyceric acid to 3 phosphoglycerate. This is catalyzed by the enzyme phosphoglycerokinase. Phosphoglycerate kinase also you can call it as. Fine. Here also how much energy is released? 2 ATP is being released. Next, 3 phosphoglyceric acid to 2 phosphoglyceric acid. Okay. This is by phosphoglycerate mutase. And finally, enolase is the enzyme here which convert 2-phosphoglycerate to phosphoenol pyruvate by the enzyme enolase. Here hydrolysis happen where water molecule is being removed. And the last step, that is phosphoenol pyruvate to pyruvic acid. This is by the enzyme pyruvate kinase. Okay, and here also how much energy is released ma? 2 ATP is released. Once look into all the 10 steps of glycolysis. Shuttle systems I will be explaining you when I am teaching ETS beta. Malate aspartate shuttle system and glycerate 3-phosphate shuttle system I will be explaining you when I am telling you the electron transport chain. Okay. As of now, focus on glycolysis. Okay. So, it is a 10 step reaction, glucose to glucose 6 phosphate by which enzyme? Louder, hexokinase, because if you say it you will remember it. Glucose 6 phosphate to fructose 6 phosphate by what enzyme? Phosphoglucoisomerase. Fructose 6 phosphate to fructose 1,6 bisphosphate by phosphofructokinase. This is the rate limiting step, means this will decide the frequency of the reaction to take place. How fast glycolysis have to take place, how slow it has to take place will be decided by this phase. And by what enzyme? phosphofructokinase. Here also how much energy is utilized for this step? 1 ATP. Next is fructose 1,6-bisphosphate to 2 molecules of triphosphates by the enzyme aldolase, and this is isomerized, so by the enzyme isomerase. Next, glyceraldehyde 3-phosphate to 1,3-bisphosphoglyceric acid by what enzyme? Glyceraldehyde 3-phosphate dehydrogenase. Okay. Next is by phosphoglycerokinase, and here 2 molecules of ATP are being released. Which step this is, ma? 7th step, and NADH is released at 6th step. Do remember. Next, 3-phosphoglyceric acid to 2-phosphoglycerate by phosphoglycerate mutase enzyme. Next, this is by enolase enzyme, this is hydrolysis reaction. Next is phosphoenol pyruvate by the enzyme pyruvate kinase, and at last 2 molecules of pyruvic acid are formed. So, here in glycolysis what you have to majorly remember is what are the rate limiting steps here. Okay, what are the rate limiting steps? Rate limiting step is 3, and 3 phases are irreversible reactions. Irreversible, those are at 3 by phosphofructokinase, at 7 and a last step, that is 10th step. Okay, at 3, 7 and 10th step. Fine. And tell me at what steps you can see substrate level phosphorylation. First tell me what is substrate level phosphorylation. Substrate level phosphorylation is nothing but phosphate group is transferred from the substrate to ATP. So, here ADP is there, right? Okay, ADP plus inorganic phosphate will give you ATP. Okay, that inorganic phosphate is taken from what? Substrate, and forms the ATP over here. Last also that is the same, ADP plus inorganic phosphate from phosphoenol pyruvate will give you ATP over here. Like that you have 2 SLPs at what steps? One at 7th step and one at 2 substrate level phosphorylation. Clear? So, now what is the rate limiting step beta? 3. What are the irreversible steps of glycolysis? 3, 7 and 10. Substrate level phosphorylation, where do you see? 7 and 10. Okay. So, are you clear with the reactions? Shall we go for energetics of glycolysis, which is very important? Yeah, energetics of glycolysis. See, glycolysis, what all points we are learning is very much important, ma. Okay. There is no wonder if we see structures also. Glucose, glucose 6-phosphate, all this we have to learn with structures as well. In next class we will deal it in the advanced way. As of now, remember each and every enzyme, what are the rate limiting steps, substrate level phosphorylations, irreversible steps and energetics of glycolysis. Okay. Now, if you have to see the energetics of glycolysis, first you tell me in what steps ATP is being utilized? First and third. In these 3 steps ATP is being utilized by glycolysis. 1 ATP. Okay. So, overall how much ATP is utilized? 2 ATP. Wherever energy is released, you tell me? 6th step first, right? In 6th step how much energy is released, ma? 2 NADH. Okay. 7th step 2 ATP. Last 10th step 2 ATP. So, here is a question, where are these 2 plus 2 4 ATP are being released, right, from glycolysis? Where are they being released? To cytoplasm only, right? If this is the cell and this is the cytoplasm, glycolysis happened, 4 ATP are released where? To the cytoplasm itself. From that 4 ATP, 2 ATP are being utilized by the investment phase. Okay, first and third steps. Okay, this energy whichever this first and third steps are utilizing have not come from space, they have come from here only. So, if we have to calculate the net ATP of glycolysis, what is the net ATP from glycolysis? You tell me. Net ATP will be 2 ATP, right? Because of the 4 ATP which is released into the cytoplasm, 2 are being utilized by first and third steps. Here comes second type of question, what is the total ATP released from the glycolysis? 2 NADH is there, right? So, here up till our intermediate, for our approximate calculation, remember ma, 1 NADH is equal to, we take 3 ATP. Okay, 1 FADH2 is equal to 2 ATP, okay, for our calculation. Accurately though, this is 2.5, this is 1.5, okay, as of now you remember like this. As such, if 2 NADH are released into the cytoplasm, how much ATP it is? 2 NADH means, very good, it is 6 ATP. Plus this 2 ATP, right? 2 are the net ATP. Total ATP is how much? Clear with the energetics of glycolysis? Fine, right? What is the net ATP and what is the total ATP? Clear? And where do you find the irreversible steps, rate limiting steps and substrate level phosphorylation? Once go through everything in a single minute, just go through it. Done, right? Net ATP and total ATP, energetics of glycolysis is clear. In whichever steps energy is being released and utilized, that should be clear in your mind. Okay? Online students, you can take a screenshot of this ma. Now, after formation of 2 molecules of pyruvic acid inside a cell, what happens? 2 molecules of pyruvic acid, if it is aerobic respiration, what happens? Enters into mitochondrial matrix. Good, Akshita. Mitochondria is how many membrane bound? Double membrane bound. Outer is outer mitochondrial membrane and inner mitochondrial membrane is enfolded into what? Cristae. Space between outer and inner mitochondrial membrane is what? Intermembrane space, and inside is mitochondrial matrix. Okay. These 2 molecules of pyruvic acid entering into the mitochondrial matrix, it will conduct 2 successive steps. First is what? Link reaction. Link reaction is also called as what? Oxidative decarboxylation. Second is what? TCA cycle, Krebs cycle. Okay. After this, complete energy is being released, that is continuing the last step of aerobic respiration, which is electron transport system or electron transport chain. Clear with all the 4 steps and the locations, right ma? Now, if we have to look into anaerobic respiration, everything happens inside the cytoplasm only, no? I'll tell you, 1 minute. So, here if you have to see, these 2 molecules of pyruvic acid inside the cell, if it is conducting lactic acid fermentation. Lactic acid fermentation, the best example is what? Lactobacillus. Okay. And the second type is alcoholic fermentation. Alcoholic fermentation takes place where? By yeast cells or brewing, those bacteria which is used in the brewing industries, okay, for making duffs and all. So, those will conduct what? Alcoholic fermentation. At the end of lactic acid fermentation, only lactic acid is formed. And in eukaryotic cells also, like you know, muscle cells of eukaryotes, these cells, whenever we do vigorous exercise and all, okay, then the cells will demand, then the cells will demand high oxygen, okay, whenever we do vigorous exercise, cells will demand high oxygen. So, when they are deprived of oxygen, they will conduct what? Lactic acid fermentation for immediate release of energy, okay. At the time of conduction of lactic acid fermentation, at the end what is formed? Lactic acid is formed. Because of the accumulation of lactic acid inside the muscle cells, they get muscle cramps after vigorous exercise. That is the reason in eukaryotic cell, fine. So, now if we have to see clearly alcoholic fermentation, till here we know, okay, from glucose to pyruvate. This is first converted to acetaldehyde and then to ethanol. Ethanol, and here acetaldehyde and carbon dioxide is being formed plus CO2. So, here you can see carbon dioxide is being released. So, at the end you have to count what are the end products of alcoholic fermentation, ethanol and carbon dioxide. So, here if you have to see NADH, whichever is released in the 6th step of glycolysis, is utilized by the alcoholic fermentation as electron acceptor, okay, as electron acceptor to give out NAD plus, okay, and this also is being utilized here in the glycolysis for the release of NADH. That is why for anaerobic respiration, the net energy released is 2 ATP. Complete energy released also is 2 ATP in the cell. That 2 ATP is also coming from where? Glycolysis, not from fermentation. Clear? Alcoholic fermentation is clear, right? What are the end products? Carbon dioxide. Where does this takes place? In the cytoplasm only, and total energy released is 2 ATP. NADH, whichever is released in glycolysis, is used here for the electron donor. Fine. So, here this is for lactic acid fermentation. Only lactic acid is formed here, and here also the same case NADH is being utilized over here, and the net ATP released is 2 ATP. So, if you take a eukaryotic cell after the completion of aerobic respiration, the complete energy formed after aerobic respiration in a eukaryotic cell is 36 ATP. Complete energy is formed how much ma? 36 ATP in a eukaryotic cell. Fine. In a prokaryotic cell it is just 2 ATP in a prokaryotic cell. Fine. So, this is less than 7%, yes or no? The energy released in a anaerobic respiration process is less than 17% of aerobic respiration. If you have to see alcoholic fermentation meta, alcohol more than 13% in a cell will make the cell poisonous. Okay, how much percentage? More than 13% of alcohol, that is ethanol, inside the cell will make it poisonous. Fine. These 2 points from our NCERT, we have to remember them. Next. So, yes, this is all about glycolysis and anaerobic respiration. Any doubts anywhere? Okay. And there is one more question. If there is a question like in aerobic respiration and anaerobic respiration, glycolysis is common, we all know that. Glycolysis is common in aerobic and anaerobic respiration, we all know that. Glycolysis in aerobic respiration will give how much of energy, total energy? Glycolysis for anaerobic respiration will give how much energy? Remember that. 2 ATP is the total energy here. Here how much is the total energy? 8 ATP is the total energy. What is the net energy? That is it. Okay? This is the way you have to learn. Now, shall we exercise few objective, okay, from glycolysis and anaerobic respiration? Look into it. Online students also answer ma. reaction of glycolysis where a water molecule is removed. Hint, last but second step of glycolysis. Hydrolysis నందు ఎక్కడ అవుతుంది? 2-phosphoglycerate to phosphoenol pyruvate. Correct ena? Yes? Good. This is the last step. Okay, this will be the first step, and this is the fructose 6-phosphate to fructose 1,6-bisphosphate is the third step, rate limiting step. Okay, third step. It is by what enzyme? Phosphofructokinase. Okay, next. Less than 7% of energy. Good. Next, this is from 2022. ATP is used at 2 steps in glycolysis. This assertion is true only. Reason, first ATP is used in converting glucose to glucose 6-phosphate, correct kada? Next, and second ATP is used from conversion of fructose 6-phosphate to fructose 1,6-bisphosphate, true, right? So, both assertion and reason is true. Reason is explaining where ATP is being utilized, right? So, here this is the correct explanation. Whenever assertion and reason are true, you just look into whether this is explaining assertion or not, then you can decode it. What is the net ATP? Net ATP when each molecule of glucose is converted to 2 molecules of pyruvic acid. Net adugutunnadamlo 2. Okay, next. Last, locations we have discussed. Citric acid cycle is nothing but TCA cycle or Krebs cycle. Happens where? TCA cycle. Matrix, right? Good. Glycolysis? Good. ETS? Inner mitochondrial membrane or cristae. Proton gradient, that is ETS. Okay. Electron transport system channels will be in the inner mitochondrial membrane, but proton gradient is from intermembrane space. Okay. So, now answer me properly. Once again for your reference, I will tell you, see, if you consider this as outer mitochondrial matrix, okay, and enfoldings, just roughly I am drawing, this is inner mitochondrial matrix, space in between that is intermembrane space, and this is what? Mitochondrial matrix. Here what happens once the pyruvic acid enters, what happens? This gets converted to 2-acetyl-CoA, okay, by what? First is oxidative decarboxylation or link reaction. Second is good TCA cycle, okay. After that, in intermembrane, inner mitochondrial membrane, what is present? Electron transport system. Proton gradient is generated in intermembrane space. Clear? Clear beta? Now answer the question properly. Second, A is 2, B is 1, C is 4, D is 3. Option 2 will be the correct answer. Fine, understood, right? So, yes, tomorrow we will start with another chapter, okay, and we will build the foundation of that chapter as well. Fine, thank you.